Hello, you are watching single-cell multiome analysis series with SciDAP. In the previous video, we started the QC analysis of our aggregated sample. Now let's take a look at how the data looks like after the filtering. Open your analysis and first navigate to the Unfiltered tab. Here you could find the plots picturing different characteristics of your data. The first two PCA plots reflect how the metrics themselves correlate with each other in your sample. For example, it is expected that ATAC fragments in peaks and the number of peaks have strong correlation, as well as RNA reads and genes. They would correlate well in both plots if the data is of a good quality. These PCA plots help to understand whether metrics could have a joint effect and how changing the threshold will modify your data on multiple parameters. If we switch to Filter tab, you could notice that PCA plots slightly changed. You could enlarge them in your browser or right-click and open them in a new tab for the better view. Moving forward, there is a bar plot showing cell numbers for every sample in the dataset. In our case, we observe a disproportion of cell numbers between the samples. There is a plot called Distribution of RNA reads per cell. I prefer to look at this kind of plots in the Unfiltered tab because we could see the distribution of the entire dataset, and it is shown where we put the threshold. If you remember, it was 1000. So you could see that for some samples, it is perfectly cuts the population that is probably low quality and includes a major population of cells and within the distribution of gene per cell. As you remember, on the PCA plots, these two metrics are correlated. And here we've set two thresholds, the bottom and the top one. We could see that the 250 as a minimum number of gene per cell probably is too far from our major peak here. When we will be rerunning the analysis, we might want to move our bottom threshold closer to the major peak. And the maximum number of genes per cell was set as a 5000. It also seems that this characteristic was set as too relaxed, so we could make it more stringent, make it maybe 3500, or try 4000 first and see if that would cut enough of outline population. This plot actually shows how these two characteristics correlate with each other, with all three thresholds put on the single plot. So you could probably see, maybe I should enlarge it a little more. So you could see, if we set the threshold more stringent, how big of the proportion of the cells it would cut from our sample. The colors show the percentage of the mitochondrial genes. So you could see that those cells that have very low number of genes per cell and reads per cell are more enriched with mitochondrial genes as well. It probably is a result of the contamination, so we want to make sure that this population is not included in our analysis, or it's set to minimum. And here it is also showed the RNA reads versus mitochondrial percentage per cell, where we set the mitochondrial percentage to 10 as a maximum allowed in our dataset. This is just the distribution of RNA reads to mitochondrial genes per cell. It shows you the maximum percentage of mitochondrial genes allowed at our data. So if we would make it more stringent, it would probably affect the sample because it's already cutting some of our major peak. There is a similar plot for the novelty score. We could see it is quite far, so we may set it a little bit higher. Remember that some metrics could overlap, and when you set up the threshold higher, for example, for RNA UMI, This population may be not present after the filtering. This is the distribution of ATAC fragments in peak per cell, and you could see that our samples are very diverse. Some of them don't have major peak at all. Maybe there was an issue with the sample prep. However, we don't want to filter too much of the sample. If we will set that higher when we lose the population on this green sample. So we can return to this metric later if needed and fix it. So there are more metrics that help you filter the low quality cells, some of them related to blacklist regions, some of them related to transcription star size score, some of them related to doublet. Additionally, there is a plot showing all metrics together on one plot, all before the filtering, just to have an idea where your thresholds would be better to set. And on the filter tab, you could see all the same characteristics on already subsetted population. There is also a tab which shows you the filtered samples split by group. Remember, we set two conditions, so we could compare our recirculating cells and tissue resident cells. If we wanted to see how they might be different in terms of different parameters. And additionally, you could download any of the files that were produced using this pipeline, including the PNG of all the plots that were shown in this tutorial. If you are ready to rerun the sample, you can go back to Overview, click Edit, and change the parameters here. However, if you would like to keep this sample for future reference, you could clone the sample, give it a name that would be different from the previous one. You could also specify, for example, here on the details, what exactly you are changing in your new sample. And you could modify everything here. Alright, now we click Save and wait until it's done. And in the next video, we will quickly review the results and move to the next step.